Now, as a first presentation, we will hear from Professor Dirk Bumann from here in Basel, the topic of anatomical and cellular barriers for targeting pathogens in rodents and human tissues. So the floor is yours.
Yeah, thank you, Patrick. Good morning. I thought I present to you some of our insights into accessibility of bacterial pathogens in tissues, because that is an obvious, I think, concern for anything larger than small molecules. So if we just look at any human tissue, it's very complex, has different compartments, has barriers, and during inflammation there will be even more barriers. So the question is this of concern to, or is it difficult to reach the bacteria in these complex tissues? And this is actually pretty much unknown. It's amazing, after 120 years of visualizing bacteria in the microscope, we don't know for almost any infection where the bacteria are actually, in a comprehensive way. This is one of our interests in our group. And I will present you shortly two different projects. One is a model. It's a Salmonella infection model. This is a model for typhoid fever in mice. And we also work on Staphylococcus aureus, a gram-positive bacterium in humans. So we look in these joint infections. So starting with the mice, it's, the challenge is really to find submicron particles in centimeter-sized organs. It's a severe challenge. And we look for ways to overcome this challenge. And one of these methods comes from neurology. They want to follow axons across entire brains. These also axons are very thin, and they came up with this method that's it's called serial two-photon tomography. So what you do is you have your tissue block, you chop off the surface, and use a two-photon microscope to scan the entire surface at submicron resolution. And then you chop off another slice and you rescan the newly emerging surface. So overnight you can eat through an entire organ, and you have then about 1 terabyte of data. And what we see then, this is like a 3D animation of this data set, of such a data set. And here we also stained a marker for a component of the spleen called marginal zone that's in between two compartments, red pulp and white pulp. So it's that the details are not important for you, but we have two major compartments in spleen, red and white pulp. And we can, in the data set that we have in these 1 terabytes, we can find the bacteria and localize them in either in the red or white pulp. And the funny thing is, if we treat infected mice with fluoroquinolone, a commonly used antibiotic, or a cephalosporin, another commonly used antibiotic, what we see is that it's easy to eradicate, or at least clear, most of the bacteria from the one compartment, from the red pulp on the left, but it's very difficult to clear them from the red pulp. See a striking difference, and you have these survivors that you cannot get rid of, and they will cause a relapse after treatment termination. And we initially thought this could be a penetration issue. And this, I think, because these red bubbles I showed you in the previous slides, this red pulp, is a strange compartment. It has some arteries going through, but there's no capillary network. So there's no direct blood circulation to this compartment. Anything that wants to get into this red pulp, white pulp, needs to go from diffusion from the outside. So, and that's also true. Similar things exist in all the lymph nodes that we have. And if you use a fluorescent dextran marker, and just inject this into the bloodstream of these mice, you will see that the white pulp is not reached. And that's because there is a filtration filter that excludes anything larger than 70 kilodalton, including antibodies. So this is a privileged site for the bacteria, because anything large will have trouble to go there. And we measured, actually, using laser dissecting microscopy, we measured the content, but the small molecules still penetrate well. Now, fluoroquinolones have good penetration, tissue penetration anyway, but they also enter this white pipe. So this was not the reason why the antibiotics don't work. The actual reason is, and another difference between these compartments is that the immune response, the inflammation, the infiltration of neutrophils and monocytes is also very bad in this white pulp. And Salmonella is a slow-growing bacterium. It needs synergy between the immune system and the antibiotics for eradication. And the synergy is lacking in this one compartment. Just one illustration. So there are compartments in tissues that are very difficult to reach with large things. Coming to the second part, this is human infections, like bone and joint infections with Staphylococcus aureus. We are very fortunate to have this pipeline from the clinics, from the surgeons and the infectiologists to us, and we have hundreds of these biopsies. Because in these bone and joint infections, you cannot cure them with antibiotics anyway. They don't, even if the strain is susceptible in vitro. They don't work. You have to have first to clean up the infection focus by surgery. You have to take out the infected tissues, and these things, what you see are examples. They are up to 1 centimeter in size. So they will cut out the infection site and then try to remodel the site. If there's too much soft tissue lacking, they have to transplant it from some other body site and so on. And for us, the question is, where are the bacteria in these sites? Again, the problem is we have like centimeter-sized tissue sections in this case, but we have to have really high resolution imaging to find the very small and bacteria that don't have a very good signal to background ratio. And we do this with a method called feedback microscopy. So we do this pretty moderate quick scan of the entire section, and then we see some things, for example, the elastic fibers that have autofluorescence in the same spectral range than our dye that we use for staining Staph aureus. But with AI these days, we feed in images from many bacteria, and then the AI can really identify areas where we have these bacteria, or it predicts that these areas should be, that these objects should be bacteria, and it will then maps them on the entire tissue section, and we can then focus our high-resolution, time-intensive, and big storage generating, requiring imaging only to those sites where the bacteria actually are. This speeds up the process by about 30-fold. So instead of 1 month for one section, we only need 1 day now. And we do find, it looks like this. You have this overview scan, and then you zoom in, and you indeed find this one yellow Staph aureus inside a host cell. Now, other examples and so on. And you see already that some of the bacteria are inside host cells. Another barrier, now you have to penetrate if you want to reach a bacteria with any anti-infective. And now we have, because this is now the pipeline that runs semi-automatically, we have thousands of Staph aureus from many patients. And we find that the proportion of intracellular, extracellular bacteria varies. There's diversity between all these patients. But there's always both, always intracellular and extracellular. And the intracellular bacteria are in different cell types. Now they have these CD60, and so these are surface markers that are present on certain cell types, and by the combination of these markers we can say that the dominant cell type is actually inflammatory monocytes. But again, you see many different cell types that all have also different penetration properties. And then we have also these extracellular bacteria. You think, okay, the extracellular bacteria at least should be easy to reach, but they are actually embedded, what's difficult to see here, in so-called nets. If neutrophils or other cells explode, the chromatin, the DNA from the nucleus is spread all over the place. You can see it more easily in this negative image of this magenta channel on the left. You see these threads of DNA, and they are decorated with the typical antimicrobial proteins, in this case calprotectin, but they also have lactoferrin and myeloperoxidase, other proteins. So the bacteria, and this is just one example, but we have hundreds of these examples, the bacteria are embedded in this meshwork of DNA proteins, which is also, I think, a sizable barrier for large things. So just to summarize, we find in these two infections, we find important circulation diffusion barriers. both infections have a large proportion of intracellular bacteria, and even the extracellular bacteria are sometimes shielded by these extracellular traps. So I think it's actually mandatory, that keeps us going. We think it's really important to know where the bacteria are and what are their contacts to understand what are the chances to reach them. This is our group and our funding. Thanks very much.
Thank you very much for this interesting talk. One problem we see that in clinical guidelines from international societies, you always have these basic standard therapies, for example, giving a penicillin for Staph aureus infection, despite you showed, and it's well known, that there are intracellular bacteria which cannot be reached by those antibiotics. So we really hope that at some point the knowledge that is accumulated here translates also in guidelines that make more sense from a biological standpoint, and they do now. So who has a question?
Thanks, Dirk. Do you see a difference between cidal antibiotics and static antibiotics in these different tissues and survival rates?
Yeah, so it's a very important question actually, because the static antibiotics, the name says they don't kill the bacteria. And it's amazing that the clinical efficacy is actually comparable to cidal antibiotics, that bacterial antibiotics that kill. Now we find in our models that there was a whole confusion, because bacteriostatic and bactericidal, these are not really clearly defined. They are defined in vitro, but if you block, for example, protein synthesis, it could be that they lack the virulence factors and are still killed in the tissue. But we find actually that static azithromycin is the most sold static antibiotic. It's used for typhoid fever. It does not touch the bacterial load at all. You keep the same bacteria, you prevent growth, but you keep the really high loads in the tissues. But the funny thing is that immune competent animals can maintain, even after treatment, the control of these bacteria. So I think the static drugs, at least in this one model, they are mostly buying time for the immune system to mount adaptive immune response that will then contain the bacteria. It's very interesting because antivirulence drugs would do the similar thing. And that's the question always: is it safe after treatment? What happens after treatment if you have still remaining bacteria?
Yeah, I must admit I was a little late and may have missed it. So you made an excellent presentation about the importance of the host-bound barriers, so cells and tissues and things like that. What about barriers that are created by the bacteria themselves, in particular having in mind the biofilm polymers, yes, which is like mucus, a very strong diffusional barrier, and on the other side, the cellular envelope, especially in case of gram-negatives. These are, from a delivery point, enormous barriers. Could you comment on that as well, these bacterial or non-cellular barriers?
Yeah. Yes. So, of course, I mean, this, for all the gram-negative, this is a key problem, to overcome the outer membrane mostly and the efflux pumps. But the biofilms have often been implied for the Staph aureus infections, because they are really refractory. But the diffusion is one problem. Probably also physiological state inside a biofilm is quite different from planktonic growth. We don't see biofilms in our tissues. They might be present on implants. We do not cut the titanium implants. It is possible to do so, and we want to do in the future. But we look only at the soft tissue. So in the soft tissue, there's no biofilm. But the gram-negative envelope is really tough. We think that the porins that were previously seen as a major entry pathway for hydrophilic compounds, they are overestimated in their contribution. We believe, at least in Pseudomonas, we have shown that if you take out all the porins, all the MICs of the efficacy of the antibiotics remains the same. That's a shock, but we have redone, not published yet, but redone the same thing in E. coli, a much more well-studied model organism, taking out all the 19 porins. All antibiotics except the cephalosporins have the same MIC. So probably the membrane itself is actually reasonably leaky. You also need only maybe 100 molecules per 10 minutes to penetrate to kill the cell. So yeah, this is a complex issue. I'm happy to discuss.
I agree with you on that point by my own experience. Also, the efflux pumps tend to be a little bit overestimated.